This is Peg Brady from NOAA Fisheries, and welcome to our 2024 continuation of the NOAA EBFM seminar series. Today we're continuing that series that was initiated back in 2017, and our main objective has been to increase awareness about ecosystem-based management, specifically ecosystem-based fisheries management, and to highlight the progress that NOAA and our partners are making to enhance the EBM strategies. We have had a great partnership with the NOAA Library ever since the beginning, and they continue to be a valuable co-host to this series. These seminars are offered every month, the second Wednesday of each month at 3 p.m. Eastern Time. And as Lisa pointed out, they are publicly accessible and recorded and archived on the NOAA YouTube channel. Following today's presentation, there's an opportunity to ask Jim, our speaker today, questions about his presentation, and also to follow up with him afterwards if the questions get lengthy. I want to thank many of our speakers, colleagues, and attendees who continue to offer suggestions about future presentations. Our next presentation is October 9th by Chris Kelbel, the new division chief for the Office of Science and Technology in Fisheries. And he will be giving us an update on ecosystem-based management activities as well as integrated ecosystem assessments and the future therein. So again, let me welcome Jim Thorson. He's our statistical ecologist at the NOAA Alaska Fisheries Science Center, and he conducts research regarding global fisheries status, spatial modeling, and life history theory. So I really want to welcome Jim and thank him for his patience. This is if people joined us back in March, this is a repeat of that effort. We had some technical issues back then, and unfortunately, unable to do the presentation. But Jim has been very patient and back with us today, and we thank him for his patience in that. So I'll turn it over to you, Jim. Welcome.
Yeah, thank you so much. Yeah, Peg and everyone. So, while I'm sharing my screen, I'll say, I started at the Alaska Fisheries Science Center about 6 years ago, and I started by taking over the Habitat and Ecological Processes Research Program. I don't lead that program anymore, but it got me thinking about how we integrate process research, so dedicated experiments or dedicated observational efforts, experimental, kind of, knowledge about ecosystem structure, function, and how we integrate that with our existing kind of monitoring programs. And so I'm going to share two different sort of directions we've gone with that. One of them is a time series direction that's more related to ecosystem-based fisheries management or integrated ecosystem assessments, IEAs. And the second is related to spatial analysis for habitat. So, sometimes I start out by kind of pointing out this distinction between prediction and inference, or sometimes it's thought of as correlative versus counterfactual methods. So I would say a lot of what we do in fisheries is correlative methods. I mean, it's exploratory modeling, exploratory regression, supervised learning, splines. In the spatial domain, we use this to interpolate between samples or to predict densities within a sampling domain, to predict under constant conditions. And of course, under climate change, we need to move from correlative methods to causal methods to extrapolate beyond the range of sampling to predict densities in space or time outside, under changing conditions. And it's widely understood that to do that prediction under changing conditions, you need experiments. You need structural models where you've got parameters that you know to be stationary, and you need a causal identification method. And so causal statistics is a growth area in statistics and science in general. Some people might know the Statistical Rethinking, the McElreath book, and there's sort of a whole bandwagon about causal statistics. And so again, one way to think about what I'm going to present is sort of trying to bring in causal statistics for fisheries. So the first one of these is published in Methods in Ecology and Evolution. It was published earlier this year. The DOI is below, and think that we can share a link for that paper in the chat bar. And the way I want to frame this is that it's essentially a structural model to do synthesis in integrated ecosystem assessments. So for more details, you can see this Methods in Ecology and Evolution paper. And kind of starting from the end, what I think we need is we need to take our ecosystem status reports or integrated ecosystem assessments. They're, currently sort of a clearinghouse for different time series of seabirds or protected species like fur seal pups. This is the one, some of the leading indicators for the Bering Sea in Alaska from the 2023 SAFE report. It might have physics variables like sea ice and bloom type, cold pool extent, and then there's a bunch of different ecosystem components in the middle that we sample. And typically these are presented as sort of a set of time series. So, like if you look at Phil Levin's kind of early IEA integrated ecosystem assessment theory papers, it's a compilation of time series, and we look at trends and how they go up and down. My claim is that we need to frame that time series analysis within an overarching model for a couple of reasons. One is that without an overarching model, we can't really agree on what is different than normal. So if we've got some variables going up and some variables going down, is that what we expect? Similarly, are some variables outside the range of what we expect? That's easier to do if we've got linkages. And third, it's easier to convince people that we know what we're talking about if we can kind of draw together linkages across all these variables. So, summarize all of our process knowledge in a simple kind of conceptual model like I'm showing here. So this was just sort of a first effort that we put together in a few weeks before the SAFE. Some linkages like sea ice, high sea ice leading to a different type of bloom and the impacts of the bloom type on large copepods, those are well understood. Other linkages here we are skeptical about, and we need to work on more. But this type of synthesis, I think, is a framing of the IEA that we're moving towards. So the North Pacific Council was positive about this type of effort. They made a bunch of suggestions. We're currently working on an extension doing forage fish. And so there's sort of a thought that we could have our IEA, our ecosystem status report, have each subsection with kind of headed by a simple kind of time series structural model. And so, what does this do? Well, there's a couple ways to think about it. One is sort of that it's a middle ground between our conventional EBFM and our simple process research studies. So EBFM often does these complex nonlinear models like EcoPath with EcoSim or Atlantis. And we can only get at causality, we can only make predictions about what might happen in the future or under different conditions if we just happen miraculously to get that nonlinear model right. And almost all these models have some deviation from what we observe, and so it's you get into this question of whether we're making causal inference correctly or not with them. Alternatively, process research often does simple experiments, experiments, and then does kind of simple linear models, regression models where they predict, if copepods if you've got more in fauna, how that might affect the stomach contents of a foraging bottom fish. And so, by daisy chaining together a bunch of these sort of process research regression models, we hope that we can get all the way from climate to fishery impacts. Those only work if all of the things you're daisy chaining together are independent. And so the middle ground we're missing is how we make kind of linear models where we can test out different structural assumptions about how variables are interdependent. And so how do other fields do this? Well, I'll quickly go through a couple cases, but basically there's this big vocabulary about structural equation models or simultaneous equation models, vector autoregressive models, dynamic factor analysis, instrumental variables. And all of these, as I'll try to claim, are rolled up within this dynamic structural equation modeling framework that I'll present here. So yeah, two views. One of them is sort of the math view, which I'll go very quickly through, and then the other one is the kind of inference view, like the case studies, how it's used. So the math view to start is that a lot of science does what's called simultaneous equation models. You've got some set of variables x, and you've got path coefficients linking those variables, which is assembled in this gamma matrix, and you've got some exogenous variation at epsilon. If we've got this model, we can figure out what the variance of your variables is and compare that with data and estimate the path coefficients in gamma. But this doesn't include lagged effects. And so the way we got at this is we started out thinking about how do we add in temporal autocorrelation, like what if variables affect one variable affects the same variables in the next time. And I'll kind of skip through this quickly, but we can compute the expected variance, this whole thing, using some linear algebra, using methods we draw from spatial analysis. And ultimately, we can end up with this sort of new statistical technique where we jointly look at a bunch of path matrices or matrices looking at the impact of one variable on another variable, look and include all of those either as simultaneous effects or lagged effects, and compute the joint variance across all times and variables and fit that model. So that's what this linear algebra is walking through. We can work this out visually, where we've got this overall path matrix that's this lower triangle. It has on the diagonal, it has all the simultaneous effects. Below the diagonal, it has lag 1, lag 2, and so on. So we can fit this all, and critically, we can specify it all with this arrow notation that I'll present in a bit. So I think that I went quite quickly through that, but I'm happy to follow up with people later if anybody's interested. Perhaps what's more relevant to this audience is, the inference that we get from these types of models. And so, before I get to that, I'll say, all of this is available in a new R package called DSEM for dynamic structural equation models. And it has this sort of set of features that I think make it pretty generally useful for fisheries. So you can have missing values. You might have time series that you only, physics variables we might get from satellites or a ROMS model, but a lot of biological variables like copepods or krill, forage fish, we only sample directly. We only have funding to sample them in every other year, or every fifth year, whatever. And so we end up with missing values, and we can deal with those missing values at the same time as everything else. You can include latent variables. You can include loops or non-recursive dependencies among variables. You can estimate initial conditions. And you can fit it as a big kind of generalized linear model, like with, you might have one variable that's account data, and another one that's measuring biomass. And the whole thing runs really fast, like on the order of seconds. So you can do big model selection. It's available on CRAN. So, there's this kind of CRAN splash page. There's different vignettes. It's kind of a minimal set of dependencies, including template model builder. And so it's certainly ready for prime time here. It has a package down website, so people, again, using links that hopefully are provided in the chat bar, people can go to this website and click on articles and browse through all of the different comparisons with other models and replicate all this stuff. I'll breeze through here. So in terms of inference, well, there's a couple ways to view it, of course. So one of them is that economists, think about problems that are a lot like what we ecologists think about. And so there's a famous kind of macroeconomic model called the Klein model that looks at relationships between wages, private wages, government wages, profits P, and consumption C, investment I. And economists have to think about this because they want to make a prediction about, what happens if the government has a higher surplus or deficit. How does that affect inflation rates? wages, profits. And so, the central bank wouldn't be able to do its work without having a structural model in which they can pretend, they can imagine what happens if they change, um, this or that, they change money supply, and how that's going to affect different macroeconomic variables. And so the simplest way that economists do that is using these sort of macroeconomic simultaneous equation models. And so we could specify that model compactly using this arrow and lag notation, where we say we've got a structural equation model, or SEM, where here it's showing corporate profits affects consumption within year with, or also with a lag of 1, and we'll estimate both those. Private wages affects consumption, corporate profits affect investment, capital accumulation affects investment, GNP affects private wages, and so on. And so we compile all those in a data frame that's this TS object, and we fit it using this dsem function, and we get out a bunch of path coefficients that represent a very high level summary of how the economy works. So in this case, um, if you increase government expenses, it's expected to increase private wages, and then private wages in turn would affect consumption. And so you can look through a path diagram like this and think about what happens if you change any of these variables, what the resulting effect is on other variables. And that's why economists would care about a simultaneous equation model like this. Similarly, ecologists think about similar things like trophic cascades. And so there's this famous case of, sea star wasting disease in California, where sea stars, as they decline, there's, I think, an ESA listed sea star, Pycnopodia, I think, that is often thought to affect sea urchin, graze on sea urchins. And, there's this separate sort of effort to think about, if there's a lot of sea urchins, they might eat all the kelp. Then there's a separate set of studies about how sea otters affect kelp. And so there's all these monitoring programs across the Channel Islands, where they've measured these 4 variables over time. Each variable, sea star wasting disease hit different sites at different times, and they've got different data at different sites. So there's a lot of missing values, but we can set it up as a big multivariate model. And again, we get out sort of estimates just from time series data that look like this, where Pycnopodia has if you have more sea urchins, they drive a decrease in, if you have more sea stars, they drive a decrease in sea urchins. And if you have more sea urchins, they drive a decrease in kelp. And both of those arrows are statistically significant. And so that's, basically a what's called a trophic cascade, where changes in sea stars propagates through 2 trophic levels to the abundance of kelp. And similarly, there's this positive impact of sea otters on kelp. So we can, again, we can summarize decades of process research about a whole ecosystem by using time series data, combining that with a structural model of what we think the impact of these variables is. We estimate the strength of those linkages, and that is a simple type of ecosystem model. Statisticians often talk about these types of things in terms of Granger causality, or using a vector autoregressive model. There's a different, there's an economist, Johansson, who I think got a econ Nobel Prize for these sort of co-integration models. And so again, it's sort of these vector autoregressive models are often used to represent ecosystems, where B here is a matrix of interactions. Yeah, and so we can fit a very simple 2-variable model here. So there's a data set from Ile Royale that's an island. Yeah, it's an island with wolves and moose. And these 2 wolves are eating the moose, and so you'd expect that if there's an increase in wolves, there's going to be a decrease in moose. But similarly, if there's an increase in moose, you think that they will drive an increase in their predator. So there's this sort of different effects. There's a negative interaction of wolves on moose and a positive interaction of moose on wolves. And so you can fit this type of predator-prey system succinctly using this arrow notation here, where you've got these lagged effects of each variable on each other variable. And if you do that, you get these time series of moose and wolves shown in the top, this sort of census of Ile Royale. And characteristically, the model has these sort of predator-prey cycles. So, like in the mid to late 70s, wolves increase in abundance and moose decrease. Wolves get so abundant in 1980 that then they drop off. They have a bad year and some of them starve or something, and moose start going back up. Um, and so forth. So they've got these predator-prey oscillations, and that's sort of what a vector autoregressive model can get at. You can compare the output of DSEM with all these other ways of fitting a vector autoregressive model, including Mars, the R package Mars, and you get the same estimated effects, shown on the bottom, where again, moose have a positive effect on wolves, and wolves have a negative effect on moose. The benefit of DSEM is that it allows you to, it's a very expressive and generic interface that allows you to fit, a wider range of models than any of these other alternative software packages. And then finally, why do fisheries scientists care? So, one of the classic debates in fisheries is about recruitment, and trying to think about, is, we've got $1 billion of ex-vessel landings for Alaska pollock and the Bering Sea. It tends to have lower recruitment when it's warm, or it has in the past had that relationship pop out. And so, there's a paradigm where maybe the eggs and their larvae starve faster if it's warm because they have greater metabolic demand, and that would lead to a direct temperature link to recruitment for this big industry. Alternatively, maybe it's temperature has an impact via its impact on forage. So when there's a warm year, the prey of small pollock are these copepods, and when it's warm, the copepods go into diapause earlier. So it's possible that when the pollock hatch in a warm year, all of their prey have already kind of sunk to the bottom and decided to wait it out. And so it could be that it's not actually that temperature is directly causing recruitment to vary, but that temperature is affecting their prey, and then their prey is affecting recruitment. And, this is important because if under climate, these two different variables might start having a changing relationship with one another. And so if we're going to make climate predictions, we need to know what the actual mechanism here is. It's also important because, we've got one team that's trying to directly measure, temperature or forecast it with ROMs. We've got another team that goes out and conducts field surveys of copepods. And so they're both, we have this challenge of deciding how we integrate these different data sets that we spend money to collect, and which of them actually matter to collect. And so in this case, we explore this sort of recruitment of Bering Sea pollock. And so to do that, we fitted this model, and then we had the comms team kind of work it up into this type of kind of glossy graphic. So in the Bering Sea, we know from process research that if there's a lot of sea ice, there's this ice factory that extrudes salty cold water and forms the cold pool here. And so if there's a lot of sea ice, there's also a large cold pool. And so there's a positive effect estimated. We've also seen in the past that if there's a large cold pool, that there's a huge impact on copepods. It's highly significant, and a weaker impact on krill, which in this case is not estimated to be significant. So in a cold year, there's relatively more copepods out in the fall than there are krill. If there's a lot of copepods in the field, we tend to find a significant relationship with that with direct stomach samples of age 0 pollock in the fall, where they have more copepods in their diet. Whereas krill, if there's more krill out in the ocean, it does not tend to have a very strong effect on their krill diet. And then finally, we can estimate that copod diets or krill diets, what effect they have on survival. And copods tend to have a positive but kind of weak, not quite significant association with survival, in addition to the spawner effect on survival. So the spawner to survival is essentially like a Ricker stock recruit curve. That's like a standard stock assessment trick for thinking about how, if there's more adults, they are going to eat more of their juveniles. There's sort of this density dependent effect. But this, on the left-hand side, this is all a kind of new way of trying to think about how physics affects forage, affects recruitment. And it allows us to sort of paint a picture about all of these um, the distal cause of climate and the proximal cause of biology, that really complements what we know from process studies over many decades. So, krill are this intermittent resource, whereas in cold years, there's a huge amount of copods out in the fall, and that shows up in their diet and ends up, being associated with good survival of these age zero pollock. Another benefit of this for ecosystem status reports is that this model is jointly estimating missing values, and so we get these time series plots where, um, things like cold pool extent in the top left or spawning stack biomass from the assessment, we've got a full time series. But a lot of the things, a lot of the biology, like euphausids in the summer in the middle, or the percent copods in diet, these are things we only measure occasionally. And so we can fill in missing values in our time series and present those time series plots to fisheries managers, as a sort of integrated picture of ecosystem linkages. So, yeah, why care? Well, we've already said, we do these integrated ecosystem assessments. We've talked about that. There's also this ongoing effort to do what are called ecological and socioeconomic profiles. So we want, for every assessment, there's this goal. Every fish that we study, there's this goal that we could have a report where we say we draw together all of the ecology that we know affects that fish. And so the ESPs, they're called, draw together all these time series and also have experts that make these conceptual models. However, there's this sort of big gap where people don't know how to use all of the time series compiled by an ESP, and they don't know how to use the conceptual model when it comes time to actually do the stock assessment model. And so, one avenue that we're following, that Cole Monahan and Jane Sullivan and Juliette Champagnat and I and others are following, is to directly take these conceptual models that we could make with stakeholders or indigenous knowledge holders, um, work with them, iteratively fitting these dynamic structural equation models. And then once we're happy with something, we can plug it directly into a stock assessment model. Um, and so that provides this sort of avenue to go all the way from process research through to our stock assessment. And then shifting gears a bit, I also want to review how these same kinds of insight can be used for habitat assessment. So um, there's a paper in review that I've been doing with Sean Anderson at DFO, and then Pam Goddard, who's here, a contractor, works often with the Alaska Center, and then Chris Rupert at DFO. It's in review at Global Ecology and Biogeography. It's been in review actually for about 8 months, and so I'm hoping that, it's the package is up publicly available on GitHub, and I will upload it to CRAN. At some point, maybe I'll give up on I'll just go ahead and do that, if the journal continues to take a while. And so the structure for this model is, it takes a GAM. So a lot of people use generalized additive models for spatial analysis, and TinyVast, this package, takes all the stuff that you can do in a GAM, and then it adds in a structural equation model, a spatial structural equation model, and then also a spatial version of a dynamic structural equation model. So you can do all of this structural modeling among variables at the same time that you might do stuff that you'd want to do with a GAM. And so I'll try to explain, well, I'll get to a case study of why we'd care, but first I'll note that there's this package down website where I've got all these different kind of vignettes that are pre-compiled and available online that show different tasks. So people, I and others have been working on this vector autoregressive spatial temporal model, or VAST. We started working on it 10 years ago, and it's been used in, over 100, papers and, many stock assessments worldwide. And, it can be used for expanding age composition data, for looking at morphometric condition, for integrating multiple data types, for spatial factor analysis for essential fish habitat, stream network models, and so on. And so there's vignettes now to kind of show, working with Sean Anderson, my co-author, showing how we can do this wide range of spatial analysis using this sort of simple package called TinyVast. Because we are rewriting something we've been working on for 10 years, we've got a lot of wisdom about how to set it up. And so it uses sort of basic R structures to fit different types of spatial models. So we can fit aerial data, so sometimes there's these questions in the North Pacific about different salmon spawning grounds and how they're affecting one another. So here's sort of a diagram connecting all of the different salmon spawning grounds in the North Pacific, and we can fit that as an adjacency or aerial data model. We can fit a stream network model, shown on the right, if we want to fit, eel returns in New Zealand or salmon returns. And then a lot of what we do in marine fish is these sort of continuous 2D spatial domain models using what's called the SPDE method. And so that's what, VAST and STM-TMB use, is this continuous 2D method, and that's also straightforward in TinyVast. And so thinking this through, TinyVast, it will be on CRAN. It's not on CRAN quite yet. It generalizes all the stuff in VAST plus other models. So it can do these sort of causal models like instrumental variables, vector autoregressive, dynamic factor analysis, plus all of these sort of various sort of, empirical orthogonal functions like we do in climate studies. So it's really kind of this Swiss Army knife. It's like the stock synthesis of spatial analysis, where you can get it to do a huge range of different types of spatial modeling. And so, the case study I settled on to showcase this is integrating both drop camera and bottom trawl data. So for those who don't know, there's, a lot of the crown jewels of NIMS, in my view, is these sort of bottom trawl monitoring surveys that we've done with a fixed design and fixed gear over many decades. We get we pull fish up on deck, and then we sort them to species and weigh them, and we get stomach contents and ear bones and lengths, and this is how we do stock assessment with these data. Increasingly, there's sort of a concern that, bottom trawls, we can't sample in untrawlable habitat. And so one technology is shown here on the left, where my colleague Kreshmir Williams is I think stooped over this towed camera here. So it's sort of towed or pulled on a drift behind the vessel, taking camera stills where we can pick out individual fish, get them to species ideally, and measure their length. And we can do that in both trawlable and untrawlable habitat relatively cheaply. So how would we integrate these different data sets? Well, the idea is that we can have this big multivariate model. So in this case, we're going to have a density model for sponges and separately for corals. And then we're going to have and we can only measure that using the drop camera. We also have a density of flatfishes and rockfishes in the drop camera, and flatfishes and rockfishes in the bottom trawl. So that's 6 variables total. And we're going to do these different models looking at different kind of concepts of how these data relate to one another. So, the 8 different kind of conceptual models that we fit are shown here. So one of them would be essentially no link. Like, what happens if we estimate sponges and corals affecting rockfish and flatfish in the drop camera survey? but we have no link of the drop camera survey to the bottom trawl measurements of rockfish and flatfish, which I'm calling R2 and F2 in the top left. Alternatively, we might think that the drop camera is useful to measure sponge and coral densities, and that those sponge and coral densities are indicative of, that can be related to the rockfish and flatfish measurements that we get in the bottom trawl, these R2, F2. So that's the missing covariates panel. And that's sort of conceptually similar to what we do for essential fish habitat. Building this out further, I'm not going to go through each of these in detail, but you might think that the rockfish and flatfish in the drop camera has some proportionality to the rockfish and flatfish that we'd expect at the bottom trawl at the same place, but estimating some catchability coefficient representing how they scale, representing kind of local habitat. You might have different responses of sponges and corals to the two different measurements of rockfish and flatfish and so on. And so we can fit all these models and use AIC. And when we use AIC, this model selection metric, we end up selecting the bottom and second from the right model, where it's selecting, it thinks that the best model, the most parsimonious model, is one where there's a known link. There's sort of a proportionality link between the rockfish and the flatfish in the drop camera and the bottom trawl. We can look at the estimated linkages and standard errors here and kind of go through them to interpret them. So, sponges have this positive association that's significant with rockfishes. Corals are estimated to have a negative association with flatfishes, which is also significant. So rockfishes have higher density in places near sponges. Flatfishes have lower density in places near corals. The associations are relatively similar, but not quite identical between our picture of rockfish and flatfish from the bottom trawl and our picture of rockfish and flatfish from the drop camera. So these kind of purple coefficients have a smaller sign, typically, smaller magnitude, and are less significant generally than the bottom ones in red and green. And then the fish densities are proportional in the drop camera and bottom trawl. So, if we measure variation in density with the drop camera, that gives us some information about variation, density, if we'd gone out and done bottom trawling, if we'd been able to do bottom trawling there as well. So, so that's sort of the statistical inference that we get, and it's, nice to be able to get, sensible inference while fitting this sort of spatial habitat model. We can also use the model to predict the density for these species across the entire Aleutian Islands and Gulf of Alaska. And so I'm going to zoom in here on what's called Bowers Bank. So the Aleutian Islands, stretches, from Alaska over towards, Kamchatka, Russia, and Japan. And there's sort of a segment in here called Bowers Bank that's this sort of relatively shallow stretch of area that reaches towards what's called the donut hole in the Bering Sea. And so, because it's shallow, we've gone out and we've done samples of rockfish, flatfish, corals, and sponges using the drop camera. But for whatever reason, it was never included in the bottom trawl survey, to sample Bowers Bank, even though it's shallow and it probably has rockfish and flatfish. And so we're here seeing whether we can predict variation in what the bottom trawl would see of rockfish and flatfish in Bowers Bank, even though we've never been there, and just using information from the drop camera survey that goes there. So the green, here I'm showing the predicted density of corals and sponges in Bowers Bank. There's some sponges well off in the north, corals in different spots in the kind of southern portion of Bowers Bank. And those are then used to inform predictions of rockfish in the bottom trawl in the bottom right, in the bottom row, places where rockfish or flatfish are expected to be if we'd done the bottom trawl, even though we've never done a bottom trawl survey in that area. And we can also get standard errors. We can use this to prioritize our sampling design. So again, why would we care about these types of multivariate models? Well, in my experience, people, VAST has been, very successful, I think, at, a number of things, a number of uses in fishery science. But the multivariate models in VAST have not seen as much uptake because I think they're just really complicated. And so, on the one hand, TinyVAST provides this new, expressive and clean interface to specify multivariate models that satisfy the specific scientific goals that the user has. And I'll say that there's, initial users. I mean, people are welcome to reach out to them if you know them. John Ream is using this to do standardized abundance indices for forage species. And Jenny Bigman's looked at this for looking at age-specific climate responses, and Cheryl Barnes and Perry Gerson are looking at predation landscapes in the Gulf of Alaska. So um, I think all three of these groups are people that, understandably, I think were reluctant to use VAST for multivariate modeling because of concerns about it being, a lot to a lot of detailed software to learn. And my impression is that they've all found it fairly straightforward to pick up TinyVast. It looks in software, it looks a lot like fitting a GAM or STM-TMB, and so it should be familiar to anybody doing spatial analysis. So I think that's it. I'd be happy to talk more about these or take questions about where we're going with these projects. But again, both of these packages are available online and have these sort of packaged down websites that show all of the vignettes. My email's there, and yeah, thanks to the organizers, including Peg and Lisa.
Thank you so much, Jim, for your presentation. And everybody, I did add some links to a couple of the things that Jim showed us today in his presentation. So take a look in the regular chat. And meanwhile, we've got lots of time for questions. If you take a look to your right, you'll see a separate Q&A chat where if you enter your question, Jim will answer them live. So go ahead and take advantage of this opportunity to do that. Remember that your first name does show up with your question as it goes across the screen. So if you'd rather not have your name shown, you can go down to the bottom and click on anonymous, and we'll still get the question, but we won't know who you are. So let's give it a couple of seconds to see if we have any questions. Meanwhile, I also want to remind people, I think a few people might have joined us a little late, but we are recording this, and I know that some people might want to watch this later or couldn't join us today because of time zone differences. So I'm going to add the NOAA YouTube channel link here, and in about 2 to 3 hours, I will have the recording uploaded. I got a question. Give me 1 second. Okay, first question from Jen. Are there plans to try to validate model predictions with field surveys?
Yeah. So thanks for asking, Jen. So certainly the example of the sponge and coral work that I described in this spatial case study, that builds on work that the Alaska Fisheries Science Center has been doing for, I don't know, 12, 15 years. So, we first started doing, we first fitted SDM's spatial distribution models to, I'm going to get some of these details wrong, but I think this is generally right, that we fitted them to, kind of opportunistic data we had and made species distribution models to predict sponge and coral densities. We then got money from the National Coral Program to do drop camera surveys in the Bering Sea and use those to, as you say, to validate these sort of SDM predictions. And generally, the SDM predictions did correspond fairly well to what we saw in the field after we did it. And then again, in this more recent cycle with the National Coral Program, people have been doing drop camera surveys in the Gulf of Alaska and Aleutian Islands, and I know less about how their prior modeling has come out in terms of validation. But so short answer is yes. And I agree with the kind of implication of the question, which is that it's useful to take a model and validate it. I guess I tend to, coming from stock assessment, I tend to think that, it's useful to both validate the model and also to continue to integrate new data. So, the goal ultimately is not just to fit a model and then validate it and have this binary yes or no, but to actually collect new data, figure out where the model's deficient, improve the model, and proceed kind of iteratively that way.
Thank you. We've got another question from Andrew. Let's see if I can. My technology is not being cooperative. Andrew asks, "Hi Jim, what exactly do the numbers represent in the fluxes on your food web type figures? What are the units? Sorry if I missed it. We had a power cut partway through here."
Yeah, so that's a great question, and I agree that any plot without explicit units in ecology is is worth wondering about. Yeah, so there are different figures that I showed. The spatial ones were all these log link, and so the units in the spatial analysis were like, if you had a 10% increase in sponges, you'd have, what it's predicting what percent increase in coral, rockfish or flatfish you'd expect. In the food web plots, um, again, it depends on the sort of link that you're using. So like the physics one, um, it would end up being in if you had 100,000 square kilometers more of cold pool, what would you expect the percent increase of copepods to be? And I'm just not going to remember the exact details right now.
That's fine. We have another question. This one is from Elizabeth, who asks, "Can tiny vast estimate causal links at different spatial scales? Example, global climate driver versus local habitat influencing density."
Yeah, I love this question of spatial scales. The two kind of low hanging fruit answers. One of them is, we've done a lot of research with these sort of spatially varying coefficient models. So you can have you can put in local temperature as a covariate and look at this sort of local scale effect. And then you can also put in some feature like average regional temperature and estimate a spatially varying link to that. And that's sort of the other extreme of having a totally non-local effect. And so both of those, both that fully local and that totally non-local, you can fit jointly, and that's quite straightforward. And we and others often find that you need both local and non-local teleconnections. Both have explanatory power in a complementary way. Current research that we're writing up is estimating sort of the spatial scale, of linkages. So, if you think about a fish, it's responding not just to habitat right where you caught it, but in habitat where they've been in the last month or so. And so, we're doing these models where you estimate sort of a spatial kernel of, the covariate. And, Sean Anderson and I are wrapping that up currently. So if anybody's interested, we could share the draft paper for that. It's not in Tiny Vast though.
All right, thank you. Jen came back with a comment. "Cool. I'm glad to hear that cooperative work is happening. The best work happens when models and field folks are working together."
Yeah. Yeah, I 100% agree. And the goal, again, I mean, I want to make a kind of cartoon argument here, that oftentimes a lot of process research, is done on very detailed pieces, and so people get, rating papers over a decade. You might get a sense of how you expect cold pool to affect ecosystems in the Bering Sea, for instance. But the problem is that newcomers don't necessarily have 10 years of experience in reading those papers, and so we want some model that captures kind of a bunch of different linkages. And then the other issue is that sometimes these linkages break down, and then if you're only drawing it from published papers, there's sort of a file drawer problem, and people get out of date. And so, for instance, there's this sort of long-term studies suggesting that large copepods in the Bering Sea are good for pollock. However, in 2018 was a warm year with few copepods and the biggest recruitment, egg zero year class of pollock in the Bering Sea. And so, we're drawing together this forage fish ecosystem status report using DSEM. And kind of, it's interesting that, even among very well educated people, we're not all aware of this sort of new information about, you know, this anomalous sort of 2018 year. There's sort of benefits of the field expertise to the modelers, and there's also a benefit of the modelers in sort of summarizing things back to the field expertise, and you need information going both ways.
Awesome. We did get another question from Anonymous. It asks, these packages have so much value. Thank you for the very clear presentation. Will you be offering any short courses on DSEM or TinyVast?
Well, that's very nice to say thank you. And yeah, I would love to work with people to figure out how to do that. I've done like at the Northeast Center, I did a workshop on DSEM, and feel free to email me if you've got sort of an idea, a suggestion. We did one on VAST, I think, at NSAW. And of course, now I wish I'd done that on TinyVast instead, which I think is much easier to use. So yeah, please reach out.
And just to emphasize, your email is james.thorson@noaa.gov.
That's it. Yeah, thank you.
This next question is from Sean, who says, you said the models have been applied to EFH. Can you tell us a little more about that? Has it been used to help determine what habitat to designate as EFH?
Oh yeah, and I'm sorry, I'm probably talking too casually. That certainly, species distribution models have been used for essential fish habitat designation. I think one of the first studies was Katie Achave doing salmon models, in 2012 that went into the salmon North Pacific FM Fisheries Management Plan, and then more recently in 2017, and then again in 2023, as maybe depending on which Sean this is, maybe maybe um, that my understanding is the Pacific Islands and the Northwest Center maybe have been using SDM TMB for designating EFH, and TinyVast again has all the same functionality as SDM-TNV, or a lot of the same. It's not quite as well documented, but it would allow us to explore sort of a couple EFH questions. So one of them would be what to do with species complexes, where you only can identify species occasionally. Another one would be looking at kind of age structured, so like a lot of essential fish habitat models look at juveniles and adults, and we could do that jointly, or we could do, well, so there's a bunch of ways that multivariate models might be relevant to EFH. Another example is, essential fish habitats require, it's supposed to designate, EFH for forage species. That's like component 7. And we don't often have good sampling of forage species. And so, John and Cheryl and others are looking at predator stomachs as a sampler, predators as samplers for forage. But you often have to combine across predators to do that. And so that's a case where these sort of multivariate spatial models, I think, are really helpful to get at forage availability for potential use in EFH.
Excellent. it looks like that was the last question. So, Peg, did you have any last comments before I conclude?
No, just a big thank you to Jim for his efforts, and of course to Lisa and her team at the library for providing all the support. Again, just join us on October 9th. Chris Cabell will be speaking, and we'll be sending out notices. And of course, this recording, as Lisa pointed out, is available in another couple hours for folks who may have joined us late. So thank you so much.
Thank you. And Jim, thank you for your fantastic presentation. Have a great day, everybody. Take care.
Thank you. Yep.